results in different physical properties. For example, butane has a boiling point of -1 degree Celsius and is a gas at room temperature, while isobutane has a boiling point of -11.7 degrees Celsius and is a liquid at room temperature. Another example of constitutional isomers is pentane and neopentane. Pentane has a straight chain of 5 carbon atoms, while neopentane has a branched chain of 4 carbon atoms and a methyl group. Neopentane is much more stable than pentane due to its compact structure, which results in lower strain energy. This makes neopentane less reactive than pentane. Constitutional isomers can also have different functional groups. For example, ethanol and dimethyl ether are constitutional isomers. Ethanol has a hydroxyl group, O, attached to a carbon atom, while dimethyl ether has 2 methyl groups, CH3, attached to an oxygen atom. The different functional groups result in different physical and chemical properties of the isomers. For example, ethanol is a liquid at room temperature and is miscible in water, while dimethyl ether is a gas at room temperature and is not miscible in water. Understanding the differences between constitutional isomers is important in organic chemistry because it allows chemists to predict and explain the behavior of molecules in chemical reactions. The reactivity and properties of a molecule depend on its structure, and the different structures of constitutional isomers can result in different reactions and properties. For example, the difference in reactivity between butane and isobutane can be explained by their different bond angles and steric hindrance. Butane has a linear structure with bond angles of 109.5 degrees, while isobutane has a branch structure with bond angles that deviate from 109.5 degrees. The steric hindrance in isobutane makes it less reactive than butane. Enantiomers are a type of stereoisomer, which means they have the same molecular formula and the same connectivity of atoms, but differ in their three-dimensional arrangement. Enantiomers are non-superimposable mirror images of each other, which means they cannot be aligned with each other, even by rotating them. This is similar to the relationship between a person's left and right hand, which are mirror images of each other but cannot be superimposed. Enantiomers have identical physical properties such as boiling point, melting point, and density, but they differ in their biological activity and optical rotation. Optical rotation is the ability of a compound to rotate the plane of polarized light, and it is a property that is often used to distinguish between enantiomers. One of the most well-known examples of enantiomers is the amino acid alanine. Alanine has 2 enantiomers, L-alanine and D-alanine. L-alanine is the naturally occurring form of alanine and is used in protein synthesis, while D-alanine is found in bacterial cell walls and is not used in protein synthesis. The different biological activity of enantiomers is due to the way they interact with chiral molecules in the body, such as enzymes and receptors. Enzymes are proteins that catalyze chemical reactions in the body, and they often have chiral centers that can distinguish between enantiomers. Receptors are proteins on the surface of cells that bind to specific molecules, and they also often have chiral centers that can distinguish between enantiomers. The different interactions of enantiomers with chiral molecules in the body can result in different biological effects. For example, the enantiomers of the drug ibuprofen have different pharmacological properties.